bound in a sarcomere. It's responsible for helping cover the active site on actin, and so it helps initiate a skeletal muscle contraction. And it works with tropomyosin. All right, so what I did was I copied this part of the of my drawing right here, this group of muscle cells, this myocyte or myofiber. I copied all this and I erased a little bit of it. So we're going to talk about the steps in a skeletal muscle contraction. But before we do that, we need to develop our vocabulary a little bit more. And to understand that, we're going to have to start talking about the basics of muscle cells. And we're going to talk about it on two conditions, or two. So when we're talking about muscle cells, when we talk about muscles themselves, how are they developing over time? Skeletal muscle tissue is not very replaceable. It's not very easily replaceable. There are these cells, these helper muscle cells, so they definitely can repair things. But what I'm gonna tell you in a minute is that muscle cells aren't created or aren't built by mitosis and cytokinesis. So when you're talking about development, when you're talking about development here, we're talking about muscle cells that are fetal development to adults here, or really just post-birth. So that, how do muscle cells grow? So if this is an example of four If this is an example of four muscle cells that are developing in utero, how do these muscle cells progress over time, and what do they look like when they're born? So the muscle cells go through this process. Instead of mitosis and cytokinesis, there's literally just a version of cytokinesis, of reverse cytokinesis. And what I've done here is that this is in the embryo, or developing embryo, so very early on in the individual's life. And what I have here is a very different cell. This one on the bottom here is an adult skeletal muscle cell. When I say adult, I don't mean you have to be over 21. I mean you have to be fully formed, somewhere reasonably close to being, let's say, newborn or a year old, something like, so very, very early on in their development, individual's development. So Mrs. Gaspar's son has adult skeletal muscle cells right now, and he's not an adult. In terms of the characteristics of muscle cells, the characteristics are they're long and cylindrical, and they're very different than what I drew in chapter 3. When I was talking about cells, I talked about those globular cells with a single nucleus here. On the left-hand side, I drew a cell that had 4 nuclei. They're formed by fusion. They have a very, very well-developed series of characteristic structures here. So there is the outer membrane of the muscle cell is called a sarcolemma. There is the sarcoplasm. The sarcoplasm is not extensive because there's so much found within the muscle cell. There's so many structures there, including the SR. The SR is known as instead of the endoplasmic reticulum, the sarcoplasmic reticulum. That sarcoplasmic reticulum is one of the really most important things that is characteristic of this muscle cell. And there's two parts to it. The extensive nature of the SR, it is connected to the sarcolemma, and that is the plasma membrane of the myocyte. It has these areas of the cell of this organelle that have a couple of things here. One is there's something called a triad, and as the name would indicate, there's three things, but the triad has one thing in particular that is very, very important. It contains calcium, and I've forgotten the name, and I'm very embarrassed right now.
Don't be embarrassed.
Thank you. Terminal sister, that's it. So we talked about calcium already. We talked about calcium present in the blood and in the blood plasma. And when calcium is present in the blood plasma, if there's too little calcium, then we take it out of the bones or the diet. And if there's too much, then we either put it into the urine to get rid of it from the blood, or put it into the bones, and we reduce the amount that we uptake from the diet. So that calcium then in muscles is really important in muscle contraction, and it resides in a terminal cistern, which is a part of the sarcoplasmic reticulum, and it's found in this area of the cistern called in the area of the SR called the triad. I think the final thing is that when I talk about the muscle cells themselves being long and cylindrical, they can be several centimeters long. So these things can be a couple of inches long. They're very, very long cells. Now, what do the muscle cells do poorly? They do not replicate well. The extensive SR is responsible for protein synthesis. And we're talking about, what are we talking about for protein synthesis? Those 5 proteins of the sarcomere. So the SR is responsible for initiating a muscle contraction at the level of a sarcomere. It's also responsible for building all the proteins associated with the sarcomere, and it is really integral in the steps in a muscle contraction. Okay. The SR itself is connected to the sarcolemma, and that sarcolemma, the thing that I have right here, is simply the plasma membrane of the muscle. Okay? So there's a reasonably long vocabulary associated with the myocytes. Or myofibers. once again, myocytes and myofibers mean the same thing. So now, so before I go any further, are there any questions about what I just talked about in terms of muscle cell development, going from the way that it goes? I told you that incorrectly, or simplistically perhaps, is a better term, that all cells undergo mitosis and cytokinesis, and adult muscle cells do not. Right? They repair themselves very efficiently because they have such an extensive sarcoplasmic reticulum, but they do not make new muscle cells. So their development is this unique thing with cell fusion. And so we had embryonic stem cells starting out in the embryo, and those stem cells then converted very early on in our development. They changed from embryonic stem cells to adult cells. So now the question then becomes, what are the steps in a neuromuscular junction, or what are the steps of skeletal muscle contraction now? So the steps in a skeletal muscle contraction, and some years, if you look back at old drawings of this, some years I go very generally, and other years I go really, really specifically. This year, I don't know, we'll see how it goes. But regardless here of what happens, it starts at the motor neuron. So it starts at the motor neuron, and that term that I wrote here in blue, a action potential here, I'm going to say AP is sent to the terminal cistern. Sorry. AP is sent, not the terminal cistern, AP is sent down this terminal axon. This space right here, this is called a neuromuscular junction. This is called a synapse. Right? Between the terminal axon and the myocyte, or the terminal axon and the muscle, that neuromuscular junction. There's another word for where there's two cells that come together really close but they don't quite touch, is called a synapse. When we get to the nervous system, we're going to talk about synapses a lot. The space between the two cells here is called the synaptic cleft. So the action potential is sent down the terminal axon. So the action potential is sent to the terminal axon and causes the release of ACH. I'll no longer write acetylcholine anymore. ACH from the synaptic vesicles. So what happens here is these little blue circles here have ACH in them. That ACH then merges with the neurolemma, or the neurolemma, which is the cell membrane of the neuron. It merges with that and it releases it into, where does it release it? Into the synaptic cleft. So it releases ACH from the synaptic vesicles. And to dredge up old memories, ACH is released into the synaptic cleft via exocytosis. That's from the terminal axon. So this second clause right here, ACH be via diffusion, so it's a random process, and it's really an area of high concentration or low concentration. ACH through diffusion, ACH goes to the sarcolemma, which is the cell membrane of the myocyte, and it goes into the sarcolemma through receptor-mediated endocytosis. There's a number of different mechanisms there, but that reception, they have. We have cells that have specialized receptors for these neurotransmitters, and you don't need a lot of these neurotransmitters to cause something to happen. The ACH then moves to the SR. And what's the SR? The sarcoplasmic reticulum. And I told you a minute ago that the sarcolemma and the sarcoplasmic reticulum are really, really tied together. So once again, a little bit of your touching the sarcolemma goes directly to the SR, or the sarcoplasmic reticulum. And where on the ACH moves to the sarcoplasmic reticulum and then to the terminal cistern. So now we're no longer talking about ACH anymore. Now we're talking about the release ACH, what ACH does when it moves to the terminal cistern. ACH causes calcium to be released from the terminal cistern, and as that calcium then moves to the myofibrils. The myofibrils are stacks of sarcomeres. Calcium then goes to the myofibrils and moves to so there's an area in actin called an active site. And so calcium moves to actin and the active site. And that active site, which is covered by troponin tropomyosin complex. There's one part that I haven't talked about in terms of the sarcomere, and I just remembered it. So step 6 here is calcium moves to actin on the active site of actin, and it, which is covered by the troponin tropomyosin complex. I'm just going to use the word complex now in step 7, and it's going to say the complex moves off of the active site.
And the complex is just a pair of
Is
two
the
proteins?
the 2 proteins, right?
Okay.
So I'm going to have to put 8 over here. So the complex moves off of the active site. Now here's the part that I haven't told you about with myosin here. So now, once the active site is exposed and in the presence of ATP, so now once the active site is exposed, there's a part of myosin called the myosin head, and the myosin head grabs the active site and the active site of actin. And the active site of actin is pulled on. So actin goes along for the ride on a muscle contraction. The myosin head then pulls on the active site of actin So whenever you see the word active site, that has to be on actin. When you see the word, so the active site is at the level of the protein here. The Z discs moving close together is when a sarcomere contracted. The myosin is pulling the actin, and and the idea here, the sliding filament mechanism describes the contraction of a sarcomere. So when myosin pulls on actin, myosin can't grab something, but when it grabs it, it pulls it as hard as it can and pulls it as far as it can. The more myofibrils you incorporate, or the more myofibrils you activate, the stronger the pull is on the muscle. So if you have a little bit of pressure on the muscle, there's a few myofibrils working. If you have a lot of pressure on the muscle, there's a whole bunch more working, all the way up to its maximum potential. What is that graph called when we're talking about making a graph of muscle contraction? It's called an EMG, and it's something we talked about last week, right? And we taught in your lab. But this thing right here, let's see here, I haven't talked about the structure of myosin, and we'll talk about that tomorrow in lecture. I haven't talked about the sliding filament mechanism. I want to talk about that in a second. In terms of the filaments themselves, the term filament refers to two filaments, two proteins here, the actin and the myosin. There's two ways that the actin and myosin can cause if these are the Z discs, if my two arms are the Z discs, if you're watching football, these are goalposts, right? So it's good, right? So if the arms go together, there's two ways to do it. The first is that the proteins themselves could shorten. And the second is that the proteins themselves, these filaments, that is to say actin and myosin, can slide over one another. And so if one of my arms is actin and the other is myosin, the one that's moving is myosin, my left arm is myosin, the distance between my elbows is shortening, right? But my arms are not shortening. The length of my forearms are not shortening, they are the length. The distance between my forearms is maximal distance right here where there's any overlap. And now when we have the movement of the actin, which is this, right, over the myosin, that movement is called the sliding filament mechanism. Right? So now in this case, my elbow would be the M line, or and if you go back to the drawing of a sarcomere, you have right down the midline, there's an M line. If this is the M line, this is the actin. The actin doesn't go over the M line, right? Because then it would run into the other actin molecule. The actin goes up to, but not crossing the M line. So that's the definition of a complete muscle contraction, a complete sarcomere contraction. Right? Now, I don't have two other arms to work with, so I could have one of you guys do it too, but if someone else is on my right side, then we could replicate the sliding filament mechanism here. Okay? So, are there any questions? Yes.
I was gonna ask, so when the sarcomeres are contracted, that causes the whole muscle, obviously.
No, that's not true, right? So now, when the filaments themselves are the individual proteins, and they're part of the myocyte or the myofiber. The individual filaments are part of the myofibrils, which are stacks of sarcomeres. Now, your body, your brain can say, hey, this is not 400 pounds, so I don't need to have complete muscle contraction. I can do a little bit of mild muscle contraction. So now my muscles are contracting, gravity's trying to pull this down, and I'm resisting that action of gravity, right? If you're doing full muscle contractions, right, that sustained muscle contraction, then your entire muscle is working. But what we're going to learn about this week is that actually what happens is that one part of the muscle is working while the other part's resting, and what is it doing here? It's trying to get more ATP, and it's trying to regenerate, remove calcium from it, and you need to put the calcium back in the terminal cistern, and then you need to restart that whole muscle contraction again. So what happens here is that when your muscles are contracting, it's not the entire muscle contracting, it's one part of it, and there are these things called motor units, and I haven't talked about those yet. We'll talk about those tomorrow. Okay, so it's a little bit more complicated than that. So are there any questions about this? So I want to just switch to the laptop for a couple of minutes. Let's see. Show you where we are and what we're doing. In terms of, 2 connections done. Next question. So, in terms of the things, the schedule themselves, so we are up at the top there, well, wait a minute. We're right here. We just finished the lecture about skeletal muscle tissue. The vocabulary, I want to show you what the vocabulary is in a second and where to find that. Tomorrow we're just doing a lecture. Tomorrow's lecture is going to be really, really, really detailed, much more detailed than today. If today is a problem, and generally speaking, today people will walk out like, oh my God, how do you remember all this shit? It's hard, right? And tomorrow's lecture, I'm gonna stop and I'm gonna say, we're gonna get to about slide somewhere around 40 or something, maybe 30. And that, all of those diagrams that I just drew today here, more or less cover not everything, but a whole bunch of those first 30 or 40 slides, right? What I'm trying to do is to give you an idea of one way to study something, a really powerful way of studying. If you do that drawing that I just did today, 10 times tonight, 10 times tomorrow morning, yes, I'm serious, then the possibility that you remember all this vocabulary is much higher. In terms of where we are right here, we're in this muscular system, and I just want to show you the vocabulary. Oh, and by the way, after tomorrow, if I say it or if I forget to say it, so we're having a quiz on Monday, a week from today. Come hell or high water, we're definitely having that quiz. It's going to be on chapter 8 and 9. We're already done with chapter 8. Whatever we finish tomorrow is going to be on the quiz on Monday. If we don't finish everything on chapter 9, then we're going to finish up chapter 9, wrap up chapter 9, and go to axial skeletal muscle. Okay? What does the vocabulary look like? So here is, a number of years ago, I sat down and said, what does the vocabulary look like? Just, and if you look at this list of terms right here, now this is the, just found in your Brightspace, right? So all of these things, there's tendons and ligaments. Some of these things I haven't talked about. If you look at number 13, aponeurosis, I'll talk about that tomorrow. And if not, then the following week. We talked about fascicles. Did I mention collagen? I mentioned collagen in passing. But you have to remember, if I talk about collagen, what type of protein it is and what the function it is. There's myoblasts. I don't know if I talked about the different types of cells in the development of the embryonic development of muscles. But the sarcolemma and the sarcoplasm reticulum, we definitely talked about. The myofibers and the structure of the multinucleate, terminal cisterns, and the calcium ions. The sarcolemma and sarcoplasm, the structure of the sarcomere, we're already at 40, let's see, at the structure of the sarcomere, we're already at 47, 50 terms right here, right? The sliding filament theory, the neuromuscular junction, acetylcholine, terminal axon, endocytosis and exocytosis, we're up to 60 terms right here. Some of these, we haven't talked about some of these last terms or so, but they're easily a vocabulary list of 72 terms, most of which we talked about today from this one diagram. So if you want to start studying for the quiz and for the exam, the best way to do it is to go back when I'm going to upload this drawing, check it for clarity, and upload it today into Brightspace, and I'll show you where I do that in a minute. So it's going to be in lecture notes. I have a whole bunch of stuff in my lecture notes here, and then if you look right here, there's Unit 3 diagrams. The recordings really aren't relevant. I should just take those out. And there are 3 drawings right here. It says muscle tissue 1 of 2, 3, 2 of 3, and 3 of 3. Here is an old fashioned drawing that I didn't draw, I didn't do it, I did it the old fashioned way on the board, right here. And if you look right here, I said that this is a ligament, and so I have to take that out because that's a tendon. I looked at that this weekend, and I was like, wait a minute here, that's wrong. So here is a different view of that, but it's the same material, the same information. And this, and so my drawings today will be right here, okay? So are there any questions about what we talked about today, what we're doing tomorrow, and what we're doing on Monday?
No, sir.
And once again, Tanya, don't forget, Wednesday, what are we doing?
SATs.
Oh, okay. All right, so if you guys don't have any questions, you can take off, and I'll see you tomorrow.
You finish drawing. Yeah.